Hey, welcome to McGinty AI and our interactive platform for exploring biological quantum field theory. This cutting-edge app helps visualize how quantum physics applies to biological systems through the McGinty equation framework. So you can see on the screen, we're starting at the introduction page, which outlines the key concepts, which are, quantum behavior and biological structures, fractal geometry in biological systems, quantum coherence in DNA, and neural quantum field theory. Notice the navigation buttons at the top. They let us move between the different sections of the application. Let me show you how this works by clicking on the McGinty equation. Here we see the core mathematical foundation of our work. It's the McGinty equation. This equation uniquely combines quantum field theory with fractal dimensions observed in biological systems. The equation has two parts: a standard quantum field theory component plus a fractal correction specifically calibrated for biological structures. The parameters include biological fractal dimension, typically 1.6 to 1.8 for systems like neurons and DNA, along with mass, charge, and spin components. Notice the application buttons on the right. Let's explore the DNA applications by clicking on this button. This section demonstrates how the McGinty framework applies to DNA. The equation you see describes how quantum effects influence energy transfer along the DNA strands. We can see the predicted effects, including the energy localization, the coherence enhancement, in nonlinear transfer phenomena. These quantum properties explain how DNA achieves such a high efficiency of energy transport. Now let's jump directly to the simulator by clicking Try DNA Simulator. Here's where the McGinty AI truly shines. Our interactive simulator lets you manipulate parameters and observe quantum effects in real time. Notice the three tabs at the top. You can switch between DNA coherence, neural fields, and protein tunneling simulations. We're currently in the DNA simulator. Below the visualization area, you can adjust the parameters. Let's modify the fractal dimension slider to 1.9. And the coupling strength to 8. These represent stronger quantum effects in the DNA structure. When I clicked Run Simulation, you can see how the system calculates and visualizes the results. So in this case, we achieved a 100% quantum efficiency with those parameters. The coherent length extended to 21 base pairs, and the decoherence time is 7.4 picoseconds. So the visualization shows the quantum wave function extending through the DNA structure. This level of efficiency would represent a highly optimized biological system. So now let's try a different simulator. Let's go to the Neural Fields tab. This simulator models quantum-like states in neurons with fractal connectivity. So notice we have an additional parameter here, environmental noise, which represents decoherence effects in the neural environment. So let's set the fractal dimension to 1.6. The coupling strength to 6. And then the environmental noise to 0.2, representing a relatively protected neural network with moderate coupling. I'll click Run Simulation to see the results. The results show a neural synchronization rate of 65% with an entanglement depth of 3 layers and a coherence time of 12.8 milliseconds. This suggests that quantum coherence is sustained across the neural network, which could have implications for understanding consciousness and neural information processing. Let's finally explore the protein tunneling simulator. This simulator models quantum tunneling through protein channels, which is a crucial mechanism for ion transport in cells. So let's set a high fractal dimension at 1.8 and a high coupling strength at 7, but with moderate environmental noise at 0.4. So that's more of a realistic cellular environment. So when I click Run Simulation, we'll see how these parameters affect it. So the results show a 50% tunneling probability with a quantum efficiency of 63% and a conductance of 4.3 picosiemens. According to our model, this confirms non-classical tunneling behavior in protein channel, which aligns with experimental observations of ion channels functioning more efficiently than classical physics would predict. Notice that after running any simulation, we can navigate back to the theory sections or reset the simulation to try different parameters. So this provides an intuitive, interactive way to explore quantum biology concepts that are typically very difficult to visualize. The application connects theoretical frameworks with practical simulations, and it helps researchers and students understand how quantum phenomena might actually operate in biological systems. So each component is fully interactive. All buttons respond to clicks, and the sliders update parameters in real time. Simulations provide immediate visual feedback. So this makes complex quantum concepts, very accessible and also engaging. As quantum biology research advances, we'll continue to update the platform with new discoveries and even more sophisticated models.